normally when you see yourself in a video, we all know how it looks and sounds. So it's kind of awkward.
Yeah, absolutely. So I mean, hey, welcome everybody. So I mean, this is something that actually has been kind of a cooking actually in my mind that I need to set up, you know, a meeting. Basically, I mean, the meeting between Vania and Matias, I mean, they're kind of working on neuroscience, you know, kind of overlapping basis. I mean, to be kind of discussed, you know, how overlapping and how complementary. And so I think that kind of makes sense. So then I actually had a discussion with Ara, and Ara was kind of excited on, you know, joining this meeting. And then I thought that, you know, let's have one more, and Olli-Pekka can, you know, add, you know, value and maybe some questions, and at least, you know, spend some quality time with wine with us. So here we are. So welcome. So I'm your host, and I'll try to be quiet. And so let's move on to Vania. So stage is yours. I mean, and I mean, you can probably introduce yourself as you go a little bit. So at least, you know, Matias doesn't know, and you know, tell me, you know, what great things you have done and found your science.
Oh, I don't know. Anyway, so I am a neuroscientist. I have been doing research in neuroscience for about 4 decades, on and off, somewhere in that range. Yeah, I have probably initially my background is in engineering. I'm an electrical engineer and biomedical engineer as well. But I really don't know much about biology, but you know, I've used lots of technology to kind of tease apart a little bit of biology. And I started with doing single cell electrophysiology in rats and cats and monkeys of more than 30 years ago, and then eventually moved into human brain imaging. And we've been doing human brain imaging now for 30 years or so. But you know, I'm a neuroscientist, so I love all the technologies that are available. Neuroscience is booming in the technical dimensions that are available to us. And I'm crazy enough to try and keep my fingers in all of it in many ways. So I am the head of a center which is primarily dedicated in understanding, solving the puzzle of chronic pain and opiate addiction and opiate pain relief, the interaction between those things. My center has at least 5 different projects ongoing. And so a lot of it is collaboration, but with many other groups. And many of these studies examine cellular, molecular, synaptic circuit properties in animal models using, you know, the state-of-the-art chemogenetics, optogenetics, viral vector manipulations in rats and mice. Our studies concentrate more on humans, in literally looking at the opiate use, opiate abuse, and how they impact brain anatomy, brain physiology. I've been, for the last 20 years, my research has literally concentrated on trying to understand mechanisms of what is chronic pain. And in a sense that we were crazy enough to say, you know, nothing is known on the subject and let's study it. And basically, you know, we have now published, I don't know, probably somewhere between 50 to 100 papers in the topic. And in many ways, we have literally identified risk factors and consequences and processes involved. And a lot of this has started with just studying the patient's brain properties, and then translating it back into animal models to test the, you know, cellular, molecular underlying mechanisms. using that knowledge to translate back into human clinical trials. So we currently have at least 4 different drug studies ongoing in the lab. These are all studies inspired or derived directly from mechanisms that we have uncovered about chronic pain, and they're all different pathways, different mechanisms trying to either stop chronic pain from happening or try to reverse chronic pain or to significantly diminish chronic pain. And they're all at different levels of progress. The other large amount of work that we have done over the last 10 years has been to study what is placebo. In a way, placebo is fundamental to treatment, to any clinical treatment in a sense. It's always there, right? So we've basically published a series of studies saying placebo is predictable, placebo depends on personality and brain circuitry, and we now have models that predict how much people will respond to placebo. I need to change the room. The cleaning ladies are here and I need to get out of here. I'll go outside actually, see how that works. So that's kind of in general what we have been doing. I'm enamored with the brain in general. For me, all of this is an excuse to philosophize about how the brain works. So I'm interested in how the brain works, how perception happens in the brain. And in a sense, we are very excited recently. We think we have a whole new way of how we can, in fact, even understand or analyze brain activity from a new viewpoint, where we think we have combined psychology and physiology with each other into a unified space. So that's my latest excitement in the field. But anyway, I mean, we have my lab, my own lab. There's at least 10 students who work with me, and the extended group is more like 30 or 40 people that I work with. And there's a lot of excitement, and I'm happy doing what I do. There's a synopsis of my, I guess, scientific life in 10 minutes or whatever, 3 minutes.
Wow, delighted to meet you, Vania.
Hey, yes. I introduce you my lovely brother who is on the other side of in California, and who writes much more insightful papers about much more important topics with actual models that in fact have some reality associated with them.
Yeah, I wish that was correct.
Hey, that's an excellent kind of short introduction. And so where are the slides? I mean, no, just kidding. So basically, I mean, until, I guess Matias probably has some questions, and, maybe we all have, but my question is actually that, what is the discovery that is either, the existing discovery, what you have made that you're most proud of, or that was actually, most unexpected?
Well, I mean, look, it's very simple in a way that when we started studying patients with chronic pain, the first paper we tried to publish took about 6 years to publish. Okay, so it was a huge battle. No one even wanted to believe that we can even do what we were trying to do. Like, why are you wasting your time doing these studies, kind of thing. So, and given that, chronic pain is 15 to 20% of the world population, we now have actual theories, models, predictors, outcome measures, drug pathways. So that's probably my contribution to humanity in general, in a sense that, we can now sit back and say, yeah, we probably did something that may in fact down the line have a positive effect on humanity. And if it's, again, this is something I've said to Mika many times, if we decrease chronic pain by 10 or 20% in the world population, you know, I've done more in my lifetime than I ever had imagined I would do in that sense. So that's really, for me, the path that we have charted, and it's fun. You know, I was at a 2-day meeting, the last 2 days with the National Institute of Health, and you know, with 100 other scientists or something. And there's a real recognition by the, at least the US NIH community, that we have contributed at least these concepts to the field. And you know, NIH is putting lots of money, funding lots of other labs to try to get them to sort of at least test our concepts or extend our concepts and whatever, you know. So all of that is kind of exciting. And yes, we have established some new principles that are derived from about 20 years of research, obviously. You know, it also takes time. Biology is complicated, as Arak continues to say, that there are so many molecules in there, kind of thing, and they're all mixed up with each other. So, yeah, it's a big mess, right? So
It's a mess.
yeah.
Yeah. I mean, it's very obvious that, I mean, if you want to achieve anything great, I mean, you have to be a kind of electrical engineer. So, I mean, I fully kind of applaud for that. So, I mean, it's a great basis. So, but hey, let's move on. I mean, any questions or comments or so Olli-Pekka, Matias, or the brother. So, I mean, what do you want to ask or comment?
Not necessarily ask, but what Vania just said about 15 to 20% of the global population suffering from chronic pain. And then when you mentioned that maybe you can help sort of 10 or 15% of that, that's 200 million people that you could help.
Yeah.
So definitely that's a humanity effort, to say the least. So it's of course it's very exciting to hear you say that.
Yeah. No, you know, I mean, at least Mika knows, we really, I think, at the threshold of actually showing these benefits very quickly, in a sense. Yeah, we keep thinking that we're almost there, but of course, getting there is always hard, right? I mean, you know, the science is one thing, and then there's society, then there you know, all kinds of politics and all those things that complicate life, and actually applying any of this to society still takes lots of efforts, and who knows where it will go, right? We've done the science in a way, but yeah, on the other hand, I don't know when, but a few years ago, I made the conscious decision that, because I would give these lectures about the mechanism X and mechanism Y, and this network is connected to here, and look how this is working. And then, you know, a clinician will raise his hand, his or her hand, and say, so what? What do you have to give me, kind of thing, right? And so you need to deliver something concrete. And that's when we really started pushing to drug discovery direction, in a way. Yeah.
Yeah, I was intrigued with your hint on perception and working on the physiology, psychology boundary, because that's where we have been working for a long time. So what kind of a new insight would you be working on? Oh,
well, I mean, so this is something really fun, that this is the kind of thing, again, I think, and at least I'm very excited about, but the point here is that, you know, this is the issue comes to this Cartesian dichotomy that was probably created by Descartes in 1600 something, 50s, I suppose, in a sense. And it's interesting because Leibniz actually himself also really realized the same issue. So there is a quote from Leibniz I recently found, 1710, where he says, you know, if I have the tools to get inside the brain and start looking at all the pulleys and cogs and the machinery inside that are working with each other, I will not understand what is thinking. I will not understand what is thinking. I will understand the machinery, but that does not give me the insight into the mind, in a sense. So this is the dichotomy of the mind and the brain, in a way, right? Which is, you know, a universal sort of problem that we've been sort of grappling with. And the problem, in a sense, is that, you know, neuroscience is amazingly successful, right? We have unbelievable information about the brain properties from, okay, not from the single molecule, but complex molecules that are like receptors and channels and their interaction and the synapses and the circuits and the information content of all of that, right? And that scales up to the level of, you know, a neuron, for example, that responds to the color blue, but does not respond to the color red, right? So that's fantastic knowledge. But then all of that comes to this crisis, which says, okay, now this neuron responds to the color red. How does that translate to my perception of the color red? Right? Obviously, if I just take that neuron out and put it in a dish, that's not enough for perception to happen, right? It has to be integrated into something and to create my psychological perception. Right? This is an old philosophical concept that philosophers have been writing about forever also, right? Wittgenstein has written about it, Husserl or whatever, all of them have written on it. This is the same subject, the same issue. Wittgenstein was I like Wittgenstein because he poses this question from the viewpoint of pain itself directly, which is my direct modality of interest. What is pain, in a sense? But you can ask the same question for color. And the other way this is posed in the past is if you have a blind person, for example, a very intelligent blind person who reads about all of the neuroscience of physiology of vision, including how receptors are organized and these things are, you know, integrate into these neurons that give rise to the colors of red and blue, etc., and has this knowledge of color. Now, one day, he or she gains visual sight back and looks at the color blue. Now, obviously, that experience is dramatically different from the knowledge that she had put together as the information, that in a sense that information does not translate into the subjectivity of the experience itself, right? It is how do you integrate the subjectivity with our objective representation of information where we have no way of knowing how that information is integrated into psychology. So anyways, I think we have made some progress in these lines recently, which I'm kind of excited about, that we have some very simplistic way of marrying psychology and physiology to each other.
Okay, sounds interesting.
Yeah, one you mentioned about the framework or some sort of you know, the unified theory and all that you were super excited. So I mean, I'm sure you may not be able to reveal it yet, but I mean that you seem to be quite excited that, you know, that you're finally, you know, putting the pieces together.
Yeah, I have nothing to hide. I mean, you know, it's not, I can definitely reveal it, especially since you're going to post this thing on Facebook and have everybody watch it, right?
Yes, we're actually live streaming. I mean,
yeah. and it's a very popular streaming too, right? So no, anyway, so we have recently been looking at some meta-analysis methods of analyzing brain activity patterns. If we take about 12,000 brain imaging studies and use them in a covariation sort of type of principal components cluster analysis of some sort, and it's beautiful because we end up with essentially 6 essentially independent dimensions that are defined now by psychology and brain physiology at the same time. And so now we are just beginning to use these time. So in a sense, we reduce the brain in these brain functional imaging studies are made out of about 250,000 voxels, right? So the dimension of these studies are in that scale, and that's the spatial scale in a sense, right? And then we reduce all of that into 6 dimensions essentially. But the nice thing is that it ends up being integrated. Each dimension becomes a psychologically defined dimension, and we're now just beginning to explore how that kind of helps us with understanding brain activity, a whole new way of sort of looking at brain activity.
I mean, it's like perception and conscience and titles like that.
Yeah. So in a way, in these 6 dimensions, we kind of say that if you are in location x1, y1, z1, theta 1, whatever, beta 1, and zeta 1, then you are thinking about what Vania is saying, something like that. In a way, I'm making it a little silly, but we can say that you are using attention by this much, introspection by this much, sensory motor movement by this much, etc. kind of, and it becomes an integrated state of consciousness in a way.
So we call that the eigenmodes of the brain.
Yeah, of course. Yeah, well, that's fine. Exactly. Yes. Yeah.
Okay, sounds cool.
Yeah. Anyway, it's a new effort. We're very excited about it. We wrote a big proposal on the subject. I don't know what people are going to think about it, but anyway, it's exciting. Yeah. So,
Mika, you are muted.
Yeah, sort of mind and body, but more like mind and brain quantified type of thing in a very kind of simplistic way. But hey, excellent. So we have still maybe room for one question. If anybody has any single simple question that one I can answer in 2 minutes. So no,
it's good.
So okay, so excellent, Vania, well done. I mean, no slides, but I mean,
thank
maybe.
I'm trying very hard.
Yeah, it shows.
Well done, to say the least. Yes.
So exciting, but I mean, let's bridge to Matias. I mean, tell a bit, who you are, what you have been working on, and to me, it at least, I haven't read your papers at the same level and extent that I have done with Vania's, but I mean, it seems to me that you're peeling into the brain, using similar techniques, but more kind of the kind of in a different domain, not on the spatial domain, like functional imaging, more looking at the kind of time through the EEG and MEG imaging. But tell a bit about yourself and your research, and then definitely about the kind of a depression game. I mean, there might be something. Yeah, go ahead. Welcome.
Okay. Yeah, thank you. I started as a chemical engineer. I was hugely interested in pyrotechnics in high school. I spent like every evening in the school lab building rockets and bombs and whatever kind of bang stuff. So I went to chemical engineering school, and then in there I got intrigued in brain things. So I jumped with my cousin to the hospital where we did the very first ephemera recordings in Finland, and ephemera was just kind of invented then, and so that was interesting. Then I kind of I did some fMRI, MEG, and that was also very kind of recently invented then in late 90s in low temperature lab in Helsinki. I wanted to do some more wet lab stuff, so I went to a physiology lab and I got my PhD from physiology. So I did patch clamping and in vitro electrophysiological things, and ultimately I didn't want to do that anymore because I totally dislike killing the bats. Even though I might not be a super moral person ultimately, but I didn't like killing the animals. So we switched back to human neuroscience, and that's what I have been doing for the past like 15 years or so. So I have a group at the University of Helsinki, and then another one at Aalto University in the technical university side. So kind of our two big questions that we want to ask in the basic research side are about the brain dynamics and brain communication, and we kind of want to really get into the systems level mechanisms kind of approach, because like I was brought up in the physiology environment, and there everyone says that this molecule is the mechanism, or this synaptic protein interaction ion going through the channel, that's a mechanism, and your oscillations and your criticality are mechanisms, they are epiphenomena, and I'm absolutely sure that our consciousness has zero cares about that ion going through the channel, but our consciousness has a lot to do with the things, the mechanisms that regulate like millions of neurons to cooperate. So this is our angle there, and the mechanisms that we are looking at are oscillations. And we are now doing MEG-EEG in the non-invasive human side, stereo EEG to get into the mesoscale intracranial human recordings. And then we are now setting up a mouse lab so that we can run identical experiments in mice that we do in humans. And then in mice we can do micro ECOG neuropixels, optogenetics, and whatever. So we want to get from the spikes and spike pacing by oscillations to the oscillatory interactions that very transiently couple brain areas to each other, and then give rise to cognitive phenomena like attention or working memory or perception. So this was very difficult in MEG, because MEG, it's not trivial to source localize, and there's a lot of confounders. So we spent like 5 years trying to build a technological basis for actually doing like really big data MEG connectomics, and we got that done in 2010 in a PNAS paper. That was the very first MEG connectomics of working memory retention. And kind of thereafter we have done a few papers on how the systems level brain area interactions link the physiological processes to the cognitive processes or mental processes, and that's nice. The other side of that is the brain state. The jargon for that is criticality, and the concept is just that the brain kind of obviously it must be operating somewhere between no order or a subcritical state, or too much order or supercritical state. So on one hand you have coma or death. On the other hand you have epilepsy. It's too much order. And now this has been kind of conceptualized with sand pile models and earthquakes and kind of physical systems, but in the brain where you have no kind of hundreds of mechanisms, not just single one, and heterogeneous systems and all that. The criticality kind of science gets very complicated, but still the kind of the core is that very small changes in the control parameters of the system, like small changes in excitation, inhibition balance, connectivity, homeostasis of gain, whatever, they can cause huge changes in brain dynamics. So you suddenly have much more synchronization between brain areas, or much less synchronization between brain areas. And that kind of the interesting paper maybe on that was in 2013, a PNS paper where we showed that the fluctuations in human behavior are actually predicted by fluctuations in resting state brain dynamics. Because it has been known since 90s that human behavior fluctuates. Like sometimes you make a lot of errors and sometimes you don't make a lot of errors. And it seems that this is really caused by the individual differences in brain criticality. Like some people are just operating closer to the transition, and some more in the coma side. And like what you said about kind of then finding a purpose for all this. We did this for like more than 15 years, almost 20 years, and then also got kind of the idea that okay, now we want to make this pay off. Like really, who cares about the systems level mechanism of brain functioning? So we started to look into indications where abnormalities in brain functioning could be the underpinnings of the symptoms or actual causes of the disorder. So we have been focusing on depression for the past 5 years. We have also shown that schizophrenia, ADHD, maybe anxiety, and obviously epilepsy. They are disorders of brain criticality to some extent, or at least brain connectivity. But depression is a very interesting case. So there you have kind of a unbalance between the limbic system for emotions and executive system for cognitive control, where the limbic system goes hyperactive and that forces the cognitive system to go hypoactive. So the patients lose cognitive control of their emotions, and then the limbic system for emotion regulation kind of hijacks introspection, and you have rumination and kind of the whole palette of depressive symptoms. So we have done MEG for depression, and we actually have evidence for supercriticality in the limbic system in depressed patients and subcriticality.
Matias, I mean, I know a little bit about MEG. How much of the limbic system do you have access with MEG? I mean, isn't that all a surface events kind of thing? I mean,
we can reconstruct sources from almost all over the brain. The very deep structures, they are a problem. Yeah. So from the cingulate, from the deepest parts of insula, and from the most important.
That's what you mean by limbic. In a sense, you're not really talking about subcortical structures. You're talking about limbic cortical structures.
Absolutely, yes. Sorry, indeed, yes. Yeah, so it's important.
Limbic would be subcortex in general, right?
Sure.
So
Yeah. So the limbic and MEG, that's the cortical structures linked to the actual limbic system.
yeah,
That's true. It's almost impossible to get signals from the subcortical.
exactly. That's why I was confused. Well, so that's really fun. I mean, we actually have published, I think, the first paper with fMRI showing that the brain as a network is a small world network.
Yeah.
So I think that was the first paper as the human brain activity as a network, and it's in fact cited. It's one of my best cited papers actually. That's now almost 20 years old or so, or something like that. Yeah. So I mean, criticality and phase transitions and things like that are things that I've always been interested in. In fact, we've published papers about stochastic resonance of neuronal activity.
Nice.
I don't know, 25 years ago. So in animal experiments, kind of thing. And then, I don't know, we don't do much of it recently. I mean, we have a paper recently that we published about, anesthesia effects and brain dynamics, a really negative paper saying that all these theories don't match the data that you see in a way. So
Okay.
mostly about, resting state dynamics in rats, anesthetized versus awake. The dynamical properties are minimally changed is what we report after controlling for many, many factors. Again, that was a hard paper to publish because obviously no one wants to believe that kind of data, so. We're losing people. What are you doing, Mika?
Mika, stop.
No, I'm not doing anything. So I'm just messaging to Ara that come back. Don't go away, don't go hiding.
Yeah.
So can you? I mean, excellent kind of introduction so far, Matias. Can you actually move on to cover a bit about the depression game? And I mean, what is the kind of a approach? What do you think that and what kind of results you have actually seen so far with the game?
Yes.
And you can probably, I mean, so that you have some slides that we can. I mean, it's good to have some slides.
Yeah, I can share my screen. I really don't have slides, but I have something in PowerPoint.
Okay.
Okay, can you see me?
Yeah.
Yep.
Yeah.
Okay, so okay, let's you all know that depression is bad. Let me actually, we did this 10 years ago. We did a game that was a perceptual training game, and that was tested in a game plus placebo or game plus fluoxetine in a phase 2 clinical trial, and that was a collaboration with our colleagues and their company, and we just provided the game. And they were trying to cure amblyopia. So that's a brain disease where brain makes one of the eyes see very badly, and the eye itself is just fine. So their idea was to use antidepressant to open neuroplasticity, and then teach the bad eye to see again. And now we recruited 40 amblyopia patients, and this was the result. So blue is fluoxetine and green is placebo. And now negative changes in logMAR. This is just the visual acuity. It means that the visual acuity of the subjects is improving over the course of the intervention. and then staying improved after the intervention ends. And now this was of course bad news for the company, who didn't get to commercialize a miracle treatment of diplopia, but it shows that the game actually improves more the vision than like the game plus placebo is better than game plus antidepressant. So this was a kind of a very important result for us, showing that you can achieve huge improvements in brain disorders that are supposed to be permanent in an adult. So they ultimately regained vision of 2 lines birth in the e-shaft. So yeah, so in depression, the key systems are like executive control system, default mode system for an introspection, and then the limbic system for emotion regulation and emotional responses. The slide was created for the innovation system business people in Aalto University. So I'm sorry for the simplification. And now the push-pull mechanic in healthy brain activity tries to keep like emotions and cognition in balance, and both in contact with introspection. So this is kind of the very naive model. And now in the depressed people, the limbic system that's hyperactive, and this is what we see in MEG. So our idea was that instead of trying to do something for the emotional system or limbic system, let's try to enhance the functioning of the cognitive system, and that way help the patients regain the cognitive control. So we have done 3 versions of a game. The latest one is called Meliora. So it's a PC action game. It looks quite okay. We had some nice funding and got some game production professionals to help us implement and design the game. So we have a game first design, so that it looks and feels like a real game. But then under the hood there are plenty of game mechanics that make it a cognitive training game. And that's pretty much it. So these are just scenes from the game. It's kind of an action game where the player tries to conquer well, there is this kind of a world that has been taken over by metaphorical bad forces, and the player wants to conquer that city and make it nice again. So it becomes nice. Anyway, these are some analyses from, we have just gotten them, so they are from the latest pilot that we are working on. This is the control group. They are chronically depressed MDD patients who are not really benefiting at all from the treatment as usual. On the y-axis we have the change in BDI 21. On the x-axis we have the days from the start of the experiment, and there are essentially no changes in the BDI score in the control group or those that don't play a lot. Whereas those subjects who do play more than like more than 10 hours, we have a nice therapeutic effect. So the intervention effect here reaches the 50% decrease in the BDI score when the total gaming hours approach like 20 or no 40 hours also. And now we are applying funding to get the next pilot game done, so that we can increase retention and recruit more people. And the objective there is to achieve a 50 gaming hours experiment, and then get evidence for kind of a clinically significant depression effect, and that's it.
That's great. That's really fun. That's nice. So each dot here is a subject, or yeah.
Yeah, each dot is a subject here. Yeah.
Yeah, I mean, so, but not too many people persist on the game, it looks like, right? Which is probably the main issue with depressed people. Getting them engaged is probably half the problem, right?
Exactly. Yes.
Yeah.
Yeah.
I mean, so you know, I mean, pain and depression are highly comorbid. They coexist in a very large proportion of patients anyways, right? And in fact, we think that the brain circuits are highly overlapping with each other.
Yes.
I mean, it's interesting that your approach is from the cortical level, that you can tease out depression components. I mean, I do read the depression literature, especially with brain imaging, and that it's obviously, in a way, it's a competitive type of, I mean, I want to both learn from them and to see how we differentiate from them, what we are learning. So it's a real issue. And I am convinced that they're not the same thing, that they are uniquely different conditions, but they, at a gross level, there's a huge overlap between them. So I should look at your work more carefully, which I have not. More than it would be fun to look at. But I guess my viewpoint is more embedded from the functional imaging viewpoint, which has to do with, amygdala and hippocampus and their interaction with the cortex, and how those things are differentiated or not between chronic pain, accumbens, hippocampus, amygdala, and their communication with the cortex, viewpoint. Now there are some very recent big studies that have been real disappointments. I don't know if you've looked at them. These massive studies that were built on hundreds and hundreds of studies that they tried with huge replication, trying to identify the specific brain network for depression. And then the next study showed that, in fact, it doesn't even replicate, kind of thing. So the field is in some crisis, in a way that, at least from fMRI viewpoint, there are huge debates as to what, if anything, we have learned about depression.
Mm-hmm.
And I suspect the same will happen with pain. It's only a matter of, scaling things up and testing them more rigorously. Anyway, so so in animal models, we have shown that you can dissociate the two behaviors from each other with genetic manipulations, which are hard to understand, but they can be done, sort of thing.
Yeah. So would you see kind of a niche for behavioral kind of computerized games for behavioral treatments to depression?
Yeah, I don't know. I mean, it's interesting that you can do this. Again, I mean, the real depressed people are the ones who probably don't even want to look at the screen, right? They want to be hidden in their house, and they're isolated, and so, I mean, engaging them and having them do 40 hours of gaming itself should be a challenge, right?
Yes, yes. Yeah, but I actually, we did an kind of an interview study with like 600 something depression subjects or patients. And many of them say that they play games as a form of self-treatment. Because both for the escapism and for kind of a behavioral activation.
Yeah. But I mean, so, and what are the principles of your game besides just simply having them engaged and create some plan? I mean, is there anything more? You didn't tell us anything about your underlying the cognitive machinery that is embedded in there.
Well, it's essentially a cognitive training game, but I think it does the cognitive training part very efficiently. So it's extremely difficult. It's very well adapted to the patient, and it kind of separately targets different aspects of executive control, working memory. So we can both measure cognitive control, visual motor speed, attentional functions, multitasking. We can measure them all, and then we can selectively challenge the different aspects of cognitive functioning.
Yeah. Oh, that sounds really exciting. Yeah, I'm surprised that it is working in a sense that you can get those people to do them.
Yeah, I think
I mean, 50% change in BDI is a huge number. Although I don't know where are they starting from on the BDI scale also. So
they are very severely depressed patients. Like this, the BDI is like 27 on average as a starting point.
Okay. Yeah.
So they are moderate to severe, and I think the volunteers in our study are those patients that really are not getting any help from anywhere else.
Yeah. Well,
they have tried many drugs, and they have tried therapies, and nothing helps. So they are kind of desperate in looking for something that would help.
Well, I have a question about something you said earlier. In your stereo imaging with EEG, what is the space and time scales that you reach?
Well, in time scales, you can go down to as small as you want, like 2 millisecond time scales.
That's very long for me. Yeah. So 2 milliseconds, and then what is the space?
With EEG, you can maybe go down to some centimeters. With MEG, you can go to maybe 1 or 2 centimeters. But even that may be wishful thinking in some conditions. So it depends on whether you have something kind of specific, a response that you want to localize, or whether it's just spontaneous brain activity.
Yeah. In fact, I have a friend in Italy who does a lot of EEG studies, and we've been talking to see whether we can import this integrated dimensional space into EEG analysis, and that's something that we haven't tested it yet, but it will be something that we want to look at. I mean, in a sense that these networks that you are showing as these 3 interacting networks with these associated names, we know that these names are completely, arbitrary names. People have just
absolutely,
they're these are statistically determined networks also, right? Absolutely. There is real physiological reason for these networks to be the way they are, except for some statistical dissection in a way. And so we're trying to get away from that kind of subdivision into something more psychologically driven subdivision of the brain as a network,
Yeah.
which is how we end up with, in fact, some of them are very similar to those networks that you show, but they end up having other components incorporated into them, and then it ends up being an integrated, cognitive, psychological, perceptual, behavioral dimension in a sense.
I actually really none of the electrophysiological networks that we uncover, either intracranially or with MEG. Really none of them fits to the fMRI resting state networks that well.
Yeah.
So kind of those names and networks there on the slide were more to give and kind of to give names and concepts than as reflecting something that we would know to be the truth.
Oh. So I wonder. So your EEG and MEG analysis are based on source localization, right?
Yes.
That must be the first step in a way when you're looking at these things.
Yeah, absolutely. Yeah.
And just to tell you, source localization is an ill-defined problem that does not have an inverse solution. It has the eigenvalues cannot be uniquely identified. So you have to do lots of, hand waving to get there in a way, right? So localization with both EEG and MEG is an unsolved problem in a way, or it's an unsolvable problem in general, right?
But it is unsolvable because you don't have the necessary joint resolution, right?
No, I don't think it's a resolution issue. It's a signal mixture issue.
Well, that's what I mean. You're collecting it from much larger regions than anything fundamental underlying it, right? So
no, but in a sense, the sources are not localized, right? So in the cortex, you have signal coming from the brainstem that's mixed up in the cortex, for example. You see what I'm saying? So I mean, maybe if you look at all of them together, you can tease them apart. But
so if you can do faster than a millisecond, and you can get snapshots, let's say, that are on the scale of micron, for example, right? Then you would see it, huh?
No, I don't think so. First of all, Ara, biology does not happen faster than milliseconds, first of all, right? So this doesn't happen. It's a sluggish system, right? I mean, whatever, even a synapse is in the scale of maybe sub-millisecond, but not much more than that kind of thing. So things are happening slowly. And if you increase your spatial resolution, you end up with all these complicated molecules that are sitting on top of each other that you have to resolve that, right? So,
So hey, let's not get stuck here. I mean, it's a wonderful kind of a debate and kind of a reflection. So I mean, I think that the bottom line is that depression is a huge problem. And if neurogame can actually be built that kind of helps, I think it would be for young adults especially, it would be a wonderful way, to actually get back on track. So.
And look, if it works for depression, it might even work for pain. They are definitely built on top of similar circuitry, parallel circuitry, not identical circuitry, but there is no way, many of the depression drugs work as, sort of. If no, they don't work for either one of them, so they sort of work for both of them in a way. You can make that kind of conclusion, right? But a lot of depression drugs are commonly used for patients with chronic pain, for example. And they do something a little bit here and there.
Yeah.
I mean, out of desperation. Yeah.
Yeah, absolutely. I mean, so far I thank both Vania and Matias, and so now you are introduced. So I mean, you have a good reason to actually send kind of silly emails to each other. So I will introduce you via email after this kind of a call, and so I mean you can ask questions and reflections and whatever. So welcome. So I suggest a kind of a 1 or 2 minute break, because I mean I have run out of wine. I just need to get it a bit more. And so in the meanwhile, I mean Ara, unless you don't need to kind of pick more wine, so you can mentally prepare for the presentation of your life. So see you in 2 minutes or something like that.
Yeah, Ara, start taking some notes about your life, please.
Okay, let me look at it.
Yeah, yeah. About the inverse problem, the thing is that exactly because of signal mixing, even if you had infinite resolution, there still would be an infinite amount of possible source constellations that produce exactly the same measure distribution.
Yeah, I mean, Ara, it's very simple. The inverse matrix is ill-defined. It's just that simple. So it has infinite solution.
Yes. But then the reality is still pretty fine, because we can constrain the inverse solution to the cortical surface.
Yeah, of course.
And then the minimum norm estimates are actually pretty accurate, at least in our validations against the intracranial recordings.
Mm-hmm.
So I see the kind of inverse problem more as a theoretical problem than a practical problem.
Yeah, I see. But well, I mean, but the problem gets worse as you keep increasing the number of locations you want to solve for, right? Is that not true? I mean, and that may not be true for MEG. For EEG, I mean, at least the literature that I look at, you record from 128 channels, and then you resolve 2 sources from that 128 channels, or maximum 3. 4 is an exaggeration already. They start mixing with each other. So, so in that sense, right? I mean, that's true for EEG, at least. Is that not true? I don't know for MEG what that number is. How many sources can you actually solve for at the same time?
In MEG, we wouldn't even go for kind of exact dipole localization kind of a thing. Rather we have a source model of the cortical gray matter surface that has something like 10,000 dipoles. Mm-hmm. And we have the inverse solution for that entire surface on what kind of for each instant what kind of a current distribution you have there. And then we collapse that to a kind of a more manageable number, something like 100 or 400 parsecs of the cortical surface.
I see. We can't do that with EEG, can you?
You can. EEG is exactly the same as MEG except worse.
Except what?
Except worse. like if you had every possible information about head structure, like if you had all the conductivities right, all the layers and all meninges and whatever, EEG would be as accurate as MEG. But since the EEG source model is so bad, because the conductivities are difficult to estimate, you get the layers and resistances there, you don't get them right. Then EEG source localization is not as accurate as MEG source localization, because MEG doesn't that much care about the volume conductor properties.
Mm-hmm. Mm-hmm.
Okay, so hey, good after talk. And so, I mean, you can carry on email. So, but it seems that, I mean, I just thought that, you know, it would be wonderful if you guys actually had a chat and get to know each other. So, so Ara, I mean, time of your life. I mean, good to have you here. And so I mean, your science is kind of a, obviously, I'm not a scientist, but I mean, I have a little bit, quite deep interest on the neuroscience, and I mean, you're, you know, you have done some wonderful stuff that's pretty complicated, but I'm just wondering, how can you actually, you know, simplify it in a way that, you know, we could have a discussion on it. But let's wait for Ope to arrive. So I mean, he just wanted to have a break, but I mean, let's wait for him to come back.
Yeah.
He'll probably.
So, you know, with Vania, we have extensively discussed about the possibilities of doing a quantum treatment for the brain and the signals, and you know, the connecting the measurements with sources, etc. Right? Where again, you can end up having extreme sensitivity for a small change anywhere to generate a general macroscopic state that has these sensitivities, which is, you know, extremely well known in quantum mechanics, right? So in terms of eigenmodes of the brain, that may be possible to decompose and analyze. Right? That's a live discussion between Vania and me. We haven't completely dealt with it, but from my point of view, I always like to have more information, not less, more detailed recordings from space and time. So is Pekka back?
OP, are you online? Can you see and hear us?
Yeah, I'm online, but I have some connectivity problems, so I turn off the video because of that, but I can hear you.
Okay,
yes,
excellent.
So
go ahead.
I had some problems also. Maybe you noticed I was away for a while. I switched computers and then we had to restart computers, etc. Okay, so you know,
that's what I did.
Yeah, okay. So my you know, world is in the opposite limit. I look at the smallest and the fastest processes, and during the last 10 years, so I've been doing this for, again, 4 decades. Right? I've been involved in ultra-fast lasers applications to single molecules and microscopy, etc. And for the last 10 years or so, I was the director of a center with the title of Chemistry at the Space-Time Limit. And we meant that we basically have now the world's highest resolution optical microscope. And what that means is that we are only limited by the atomic makeup of matter. So we can see individual atoms, and we can look inside molecules with optical microscopy. And if you know, are aware of the limitations of the microscope, there is a famous diffraction limit that says that you can only see things that are of the order of half the wavelength of light. Well, we have, you know, exceeded that by a factor of about 10,000. That's what you need in order to see, you know, atoms with light. And the trick there is that what we do is we confine light. So we convert light into atomically confined quantized plasma. You know, those things may not mean very much, but you know, I'll be very maybe generalistic about it. But it is possible to actually use a silver needle. That's it, a silver needle that is atomically sharp, to essentially focus light down to 1 angstrom. 1 angstrom is now smaller than an atom. So if you have an objective lens that can focus light to 1 angstrom, you can go and look inside molecules, for example, right? And you can see them also in time. So I'm going to just share a few images to give you an idea of what we're talking about. The subject is quite general. right? So can you guys see my PowerPoint?
Yeah, yeah.
Yes.
Okay, so I'll make it full screen. So I'm interested in this ultimate limit of seeing essentially atoms move inside molecules, but it is more than a game, right? It is the limit where we're talking about now quantum sort of world of devices, measurements, and maybe even connecting to the brain, Vania. So
Okay.
do you see my cursor?
Yeah, yeah.
All right. So on this x-axis, I have space. It goes up to 100 micrometer or so millimeter scale. That's where in the Bell days, when the transistor was built, it used to be centimeters, right? And electronics basically started moving in down. Now, in electronics, we are getting to the limit of about 10 nanometers. The latest transistors are of the order of 10 nanometers. A nanometer is of the order of 3 atoms. So we're talking about 30 atoms or so in the dimensions of the object. On this y-axis, I have time, speed at which things move. So we are getting, in what's called plasmonics, we're getting down to the terahertz region. But it's not quite yet. Here I have an arrow, this red arrow, capital C stands for classical electrodynamics. Q means quantum. So there is a transition here where you can no longer think about classical electrodynamics. Light becomes matter, matter becomes light, they get merged together, and you have to deal with it something differently. So, we had proposed starting a Picoscience Institute, which is the study of things that cross this line. This is 1 nanometer. 1 nanometer is the size of 1 molecule, but we are now down to 100 picometers, which is the size of an atom. And experimentally, we are there. So we have made measurements at the petahertz time scale, femtoseconds on single molecules and single atoms. So we have, for example, I will describe to you a device, a complete device, which is an electrometer, like a voltmeter, that's made of 2 atoms. Right? Only 2 atoms, and it operates at 100 terahertz, and the localization from where we can get the signal is down to 3.4 picometers. That's crazy, right? But it's completely real, and this is a published work that we demonstrated the first single molecule device. So here are a few examples of what we are. Up top, that's the image of 1 atom. Right? This is experimental image. And again, it's done with optical microscopy, like I said, using a silver needle. And moreover, the image not only shows us the atom, but it tells us what atom it is. That's a nitrogen atom on a copper surface. That's crazy. This is looking inside 1 molecule. You see, molecules are always vibrating. They have vibrations. I'm showing this vibrational motion inside it. If molecules did not vibrate, if they didn't have springs, there would not be anything, any activity. Brain could not function if there was no molecular vibration. These springs that are inside molecules that connect to each other end up being like clockwork, right? And there will be a spring attached to a gear attached to another gear, etc. That's, if you like, the ultimate macroscopic functionality. Now, the crazy thing is that you see...
This is what Leibniz was talking about, Ara. Now you show it. That's great, yeah.
Now we show it. So you can see, this is this motion. This is a simulation. This is the real image that we have recorded. So we are seeing these bonds move inside the molecule, right? So we have a microscope that doesn't just show us the molecule, but we can go inside the molecule, right? So that's crazy. This we published last year. It's the first time that optical microscopy has reached this limit. The resolution is about 1.5 angstrom in this case. That's smaller than an atom, right? So we can see essentially the shaking, moving atoms inside the molecule, and that's nice. This is an image of the electrostatic surface, the surface charges on a molecule, one molecule, right? We can basically use this electrometer idea, or a voltmeter, to measure voltages on the surface of a molecule. And that is, again, the important shape of a molecule, as opposed to physical shape. Its electrostatic shape is what controls much of its function. The method is quite general. We can record essentially phonons. In that case, it's equivalent to seeing sound. So we can see sound atom by atom. You can see the atoms involved in, when something shakes and makes sound. So why is that important? Well, if you want to understand functional materials at the nanometer scale, you have to be able to resolve things on this scale. So we can do that. So there are, what we call the Pico Science Institute has a few aspects to it. Microscopy is an obvious one, seeing objects at the ultimate limit of what they are made of. We can make sort of single molecule devices. One molecule is a very elegant electromechanical device, right? When a molecule shakes, it has springs in it, and the springs, when they shake, they generate electric fields. So if you can measure that and manipulate that, you have essentially a single molecule device. And like I said, with 2 atoms, we show that we can have a voltmeter, complete transduction and signal, and record the signal from it, etc. And we use that, for example, to map the surface fields on the voltages on one molecule. If you have, a nonlinear circuit, which we have demonstrated also, then you have essentially the element required to make any electronic circuitry. Right? A diode gives you directional current, and if you attach diodes together, then you can generate essentially any complicated circuit. And of course, where that's going is to molecular or quantum electronics. And quantum electronics will be the ultimate limit, where people are now pushing the horizon to have a molecule essentially compute for you. We can time resolve these things, and our time resolution is in the femtoseconds, so you can see these things in life. And so there is a general concept of taking the quantum leap, but the quantum leap is really from the nanometer to the picometer scale, and we are the only people so far who have demonstrated this, or others are following. There are other examples coming out, and so this is starting to happen. Anyway, so we were trying. I'm showing you slides that were used at UCI for a proposal that we submitted to start this Pico Science Institute. This doesn't exist, by the way. It's just a proposal and what we wanted to do. So let me give you 1 or 2 examples again. This is how we look at the protein image nowadays. It's this electrostatic potential surface, which roughly defines its function. Right? We can measure that. This is purely theoretical. So now we can measure that surface. This is an example that from our data of seeing single molecules on a surface. Right? Each one of them is a molecule, but this is optical microscopy, right? With a resolution that's better than a nanometer. I'm trying to connect it to sequencing of DNA, for example, or epigenetics, etc. Well, we can see individual bonds and individual molecules. So in principle, that's completely within the range of things that opens up in biology through this technology, if you like. So the secret, yeah.
So this protein that you're showing, right? That's a model.
Yeah. So all of these protein colored images that you see, these are all calculations. Nobody's measured them.
Right? But you can see that whole protein in a way, right?
Well, so I show you the example, right? You see this one, this molecule?
Yeah.
That's the same as that image, okay?
Right.
These colors are the voltage.
But if you go to the level of to the next level. So can you see the foldings or the infoldings of the components of the protein?
So from what we have demonstrated so far, Vania, we can do a perfect job on planar molecules, atomic resolution. When it gets to be three-dimensional objects, it's a bit harder, and we're working on that. So, to go from 2D to 3D, we have to, I mean, we know how to do it, but we have to build an instrument to get there. We don't have that. So when it gets to be
uniquely resolve the folding pattern of proteins in a way, right?
Well,
which is a big problem.
Right, exactly. So our resolution is, basically atomic, right? So we're able to see it. So, I have, a list of a few things, but the key aspect of it is that we can confine light atomically, right? And if you can, have an objective that confines light atomically, then you can get into that kind of business. So single molecule devices, right now in your iPhone or your Nokia phone, what you have is MEMS, micro electromechanical devices or systems, and M is the micro. In the lab, there are developments in the nano, so nano devices that will, be in the next generation or are being introduced already. But our limit is the single molecule, right? So we can use a single molecule entirely as a device, complete device. In this case, carbon monoxide. We attach a carbon monoxide to the head of a silver needle, and that becomes essentially a voltmeter where light comes in from far away, light comes out, you just record the light, and that generates a transducer that converts electric fields into frequencies. We've demonstrated a diode type of operation in a single molecule. And I want to bring your attention to this figure up here. Do you see this? This is something from Finland. This is from the Nanoscience Center at Jyväskylä. And what you're seeing is a pyramid, but that pyramid is forged out of one sheet of graphene. Okay? These are my collaborators, and actually some of them were postdocs in my group many years ago, and we're sort of collaborating together with them. They've shown that you can take a monolayer, atomically thick graphene, and using light and local fields, you can shape it. And when you shape it, you change the electric and magnetic fields around it. So we think it's possible to take one sheet of graphene and generate from it a circuit, a complete circuit that that you can print maybe a transistor, etc., and many of them. So it's going to the ultimate limit of single atom or 2D electronics. So an example of time, this is something we published a few years back. This has even music to it. So we're looking at the breathing of one molecule in femtoseconds or picoseconds. So, it's again a fascinating possibility that we can get to the time scales of relevance. This is a collaborator, in the center where we can go to attosecond resolution of things like light fields moving around. So anyways, let me just stop this entire nonsense and say that in the limit where we are, light and matter actually become indistinguishable. They fuse. So you have to deal with this quantum world of electrons and photons becoming the same thing. And we're writing the first paper on the full quantization of dielectric in matter, etc. But there's a huge amount of possibilities and exciting world in there, which is very recent. So the first paper on seeing inside the molecule was published last year in Nature by us. And immediately after that, the Chinese group published another paper reaching the same limit. And around the world now there is excitement. And we're trying to sort of open up this new horizon in science, right? Picoscience as opposed to nanoscience. Yeah. So this is something we had published, 20 years ago on quantum logic using photons, and this is something we recently published showing that a plasmonic nanojunction, etc., can be used to essentially do quantum logic on a microscopic scale. Okay, so let me finish with the disaster. So we spent about 10 years or so in developing all of this science in a center that was funded by the National Science Foundation, but it was expected to come to an end, which it came to an end. Meanwhile, we proposed the next step in life, which is going into this picoscience, and this proposal was addressing something the National Science Foundation had initiated as what they call a mid-scale center, etc. And it had tremendous reviews. We submitted in 2019. It passed all of the critiques, etc. But they ran out of money in 2019, so they kept the proposal. They said, okay, all we need is a new budget from Congress in order to fund it. And so we waited, and in 2020, they got the new budget. So they said, patience, you're going to fund it. Then COVID happened. So NSF went into emergency spending, and the money disappeared again. So a few weeks ago, I received a declination of a proposal that had reviewed perfectly well, but essentially they ran out of money. So they kept the proposal for 2 years, and then they said no, with excellent reviews. So we have to rethink about how to do this, but we're taking a pause. So I don't know if that gives you an idea of where we are, but it's opening up a new horizon in the physical sciences, and ultimately, like I said, the synapse is made of molecules, and these molecules that come and go together have to have springs in them, otherwise nothing works. Now you say, that's too far a connection. Correct. You have to integrate. So in order to describe the function of a protein, starting from single springs, you do a lot of elegant sort of coarse graining, in order to get there. But nevertheless, it's, understanding the fundamental limit is very important. But, I think it's going to be the case that things are going to be miniaturized once more. So, the iPhone in principle, right, can be miniaturized by another factor of 1,000, right? Of course, your fingers are too big, so you have to.
That's the problem with biology, Ara. Yes,
huh?
yes. We still have fingers to use, right? Yes. Yeah.
Well, so the interface between the function of the object and the brain is another one, right? So anyway.
Yeah.
So
yeah, we just implant the phone.
Huh?
We just implant the phone in the brain, and we're done. We become machines of Facebook and Amazon. Yeah.
Yeah. Sure. Why not?
Hey, thanks, Sarah. I mean, that was wonderful. It was very engaging, and slides actually also did a great job. So I mean, it was so fun to follow the kind of a journey. I mean, brilliant. So then actually, I mean, the question is, I mean, what are we supposed to ask? I mean, the funny thing is really this Finnish connection, that you do have a strong Finnish connection. The PhDs or probably currently professors actually that actually work with you in Irvine, and so that's a sort of a funny thing.
So I have an honorary doctorate from the University of Jyväskylä. So because I've had a very old connection, I've had students and postdocs over many years come here, and in fact we just submitted I'm helping them submit a few proposals to the Finnish Academy, and one of them has to do with this graphene thing that I showed. It turns out that if you shape graphene properly, then you can generate the equivalent of a black hole. So the idea is to do a black hole type of experiment in the lab, right? You know, it's basically confining light, right? A black hole is where light gets confined and doesn't escape. I think, you know, we will probably be able to do that measurement to show that photons get trapped and cannot escape the event horizon. Roughly, that's what we are doing in, you know, focusing light down to the size of an atom. Basically, we're confining it down to an angstrom.
Yeah, so kind of a, you know, black hole in a dish. That's a funny thing.
That's exactly right.
In a little, it's a very little dish, I think.
Right, right.
Yeah. So hey, opening the discussion. I mean, you know, questions. I mean, please, Matias, OP, obviously the brother. So, what do you want to ask?
I've got a very simple question, and then maybe a more fundamental one. But Ara, can you talk about more about what you need, what you mean with the silver needle?
That's exactly what I mean. You know, what we do is we take a silver needle and sharpen it such that at the end there is only one atom. Okay. So it
That's
looks like
pretty sharp.
So that looks like the stylus of a, you know, old record player, right?
Yeah.
Yeah. So it turns out, you know, silver metals guide light. So if you shine light on it, it will guide it. If it is shaped like a needle, then it will focus it. So, you know, it will have the functionalities of an antenna, a waveguide, and a focusing lens all together if you have just a conical silver. Yeah? So, you know, the way we sharpen it, obviously, we do it under extremely high control, ultra high vacuum. We pull the needle and pass current through it, etc. But once we shape it, then it becomes an objective lens. Does that make sense, OP?
Yes, thank you.
I mean, you know,
so
we have
there is a physical.
Yeah. And of course, the challenge is that you have a lens that can focus things down to the size of an atom. Then it's just a scanner. You know, you have to have a very accurate scanner in order to be able to scan images and make images with angstrom resolution, right? So the expensive part in our lab, the hard part, is to make a microscope where, you know, what's called a scanning tunneling microscope, where we can control the position of the needle with, you know, picometer resolution.
Yeah, Matias. I mean, you know, you said that you're kind of you know, fundamentally interested on chemistry. So any questions to Ara?
Yes, yes. What kind of conditions do you need to actually use that kind of a silver needle microscope?
So, you know, in order to be able to get these images with the ultimate limit of, let's say, atomic resolution, obviously you have to, you know, stabilize a molecule. It shouldn't be moving around. If it's moving around, then you cannot do it, right? Even though there may be ways around it. So to demonstrate this ultimate limit, what we do is actually we use ultra high vacuum conditions, very, very clean environment. You know, if there's dirt, obviously at the junction, then we're going to image dirt, right? It has to be clean. And then, you know, the targeted object that we're looking at has to be immobilized. If the molecule is moving around, obviously it's hard to image it. So to immobilize it, we cool it, right? So we go to low temperatures, 5 Kelvin, right? Just to be able to get that. Now, some of the images I showed were room temperature. You don't have to do that. But of course, if you go to room temperature, then you're going to average over the dynamical fluctuations of the atoms in the molecule or the molecule itself, right? So it's a different purpose in going to low temperature versus room temperature. And
does this have the same problem as the vaccines that need to be shipped at minus 80 degrees? These new cell phones have to be at minus 200 degrees, and we have to put them in our brains.
So if the sample is small, right, let's say a few vaccine molecules, then it's easy to cool it down to minus 80, no big deal.
Assuming it needs to be minus 200, right?
It's a question of what is it that you want out of it, right? If you want to see an atom, then the atom has to be immobilized to see it. If you want to see the time average atom, then you can do it at room temperature. It's just it's the physical conditions required to demonstrate where you are, right?
Okay, so
is there any hope ever to use that in a biological system? Or
biological what?
Is there need for that?
So, in the biology, there are several applications that we really were very excited about. One of them was, I showed a picture of an RNA molecule. In an RNA molecule, you really need to know the folds, the arrangement, the secondary, tertiary structure of it, because it has charge distributions and function, which is non-trivial. So we think we can image entirely all of the properties of, let's say, an RNA type molecule, from the charge distribution to the atomic composition to etc. And will that then, translate into better understanding of genetics or epigenetics, etc.? I think so, right? It's a different level of detail. And in the case of epigenetics, the existing methods for rapid scanning and identifying, let's say, defects are really not very good. You get 30% accuracy in most cases, and you cannot really do a complete mapping. So we think that, if we decide to focus in that direction, that in biology, for example, in epigenetics, if we could make significant progress of rapidly scanning things like DNA now, right? And I don't know how much more we contribute to biology, but it would be at the molecular level of information that's sought. If the information sought is molecular, then we can do it.
Well, all right. I mean, just see if you can resolve these, even from, I don't know why RNA, I suppose, is one, like even the protein folding issues. I mean, pharmaceutical companies would be jumping up and down if we resolve uniquely these structures around which they're, you're looking for receptors or agonists and antagonists and all that kind of, chemistry that, that is dependent on all these forces, right?
Right. So let me say what is the weakness of the method at present. The weakness of the method is that we cannot operate under the biological conditions of, let's say, in aqueous solution or in the cytoplasm or a cell, right? What we're doing is we're isolating a molecule, putting it on, a substrate, and then measuring it. So that's not exactly its biological, state. Nevertheless, it's the first time that you can really measure these things, right? So, will it be possible ultimately to do it under biological conditions? Yeah, maybe but it's not where we are.
Yeah, sounds very exciting. So, I mean, Ara, when we discussed, you mentioned about, you know, the kind of a quality control for kind of a modern chips. So what was that about?
Yeah. So, you know, I can show some images, but I won't do it. But we can map out things like phonons or charge densities on, you know, surfaces, interfaces, and structures. So in the case of the latest transistors, like I said, the sizes are about 7 nanometers, and Samsung has an entire investment and a fab where they were building these transistors that are failing, but they don't have the tool to see why, right? To do process control or quality control to understand, right? Because 7 nanometers is about, you know, 40 atoms. So they need microscopy on that scale to be able to really understand what's going on, and we can do it. Right? So that part is easy. We know how to do hard materials, right? So we can not only look at the structure, but actually say what each atom is, right? Because we're measuring vibrations, so we can identify. This is chemically selective imaging, and you know, we measure current, field, charge, and space. So you know, there's no question that, you know, if Samsung wants to push the 7 nanometer or smaller technology, they're going to need the kind of resolution that only we can provide. Yeah. So we thought that that would be great. And by the way, when we were putting the proposal together, we had a significant input from the Google team in AI, right? To combine AI and machine learning in both our measurements and also interpretation of the images and the data. That's going to be another part that has to be injected for this to, you know, really be productive, right? So we can make images. Well, you know, you need the interpretation of the images, and if you're going to connect it to, you know, for example, epigenetics, you have to do another bridge, right? And also, you know, the AI part is necessary to think about because of the quantum computing aspects. Ultimately, you know, many of the quantum computing concepts come down to localizing photons, and you know, we are at the limit of what you can do with that. So, you know, we think there should be industrial interest. I mean, there is industrial interest, but we didn't develop that. We went to NSF instead, you know, thinking that NSF is, you know, the right place to launch this center, but it looks like they really dropped the ball on it. So right now, I am thinking about going to private, you know, sector for, again, redoing the essentially concept of a picoscience in multi directions.
Yeah, Ara, Opi, do you want to ask a question or reflect?
Well, yeah, it's very interesting, of course. And well, I'm a businessman. Ara is more, and you all, other than Mika, are scientists. So a businessman basically understands very little about everything. And you guys, with the exception of Mika, maybe you understand a lot about something. So it's kind of in that way really difficult to connect in terms of I understand maybe 60% of what you are saying, but I thought I understand a lot about what Vania, you were saying. I thought I understand a lot about what Matias, you were saying, and that's applicable to Ara as well. And in my mind, it's, you know, this concept about molecular vibration that Ara, you took up. Is there commonality between what you all are doing? Are we talking about chemistry or physics? Are we talking about molecules or electricity? At the end of the day, I don't know. It might be a very stupid comment. But when it comes to you, Ara, I think when it comes to, because I'm a businessman, when it comes to sort of making concepts commercial, I think we might want to have a continuation, a discussion, because there I might be able to help you thinking wise, not in practice, but thinking wise. What would it mean to because that's my business, to make things.
So I don't know what your business is. Tell us a little bit about your business.
Well, they used to say it was about consumer electronics. I would rather say it was about consumer goods, because it used to be known as a mobile phone. It's simply an item. It's an off-the-shelf item, and you basically, there's always an app that you need to do. You know, like we have this in our case, we got this measuring or microscopic application, but in fact everything a consumer buys, it's an app. A car or a dishwasher, it's an app. You buy a car to move around. You buy a dishwasher in order to wash dishes. And then you've got a lot of applications that are based on a sort of on a platform, normally a mobile phone platform, and you need to be able to perform something. So there's no real science to that, other than what I think Ara was talking about. I still think it's a B2B app, and it would be very hard to put that on a mobile phone, which is a B2C app or platform where people just don't need what you are talking about, and then in their daily life, and then it's question about how you make that a B2B app, and basically, do you need a special purpose device or platform to do it? And that's why I was asking about the silver needle, because to me it does not sound like a consumer thing. And I think, Vania, you were referring to that as well when you made your question about the temperatures, etc. Yeah. But it opens up a lot of thought, nevertheless.
Yeah. If I may reflect, so I mean, wonderful kind of science, and so I mean, my questions actually, my questions are really regarding Ara's kind of somewhat foundational, science. So my questions are really actually that what are the application areas? So Ara was actually going through some of them. So my question is really that that how far is it actually that you take the lab device or the lab experiment into something that actually could be built into a commercial device, whether that would be related to a quality control in a production line, or whether that actually would be applying that silver needle in a 5 Kelvin in an epigenetics kind of device to do this kind of mapping of basically DNA, or if it would be something else. So what would be actually all of those sounds like an areas that the value that could be tremendous, but is it more on the research side, a device that actually enables better research, basic research, or is it more like an industrial application at scale? And so I would probably pick when we discussed about this, that we would need to find a company that either Apple or Google scale, that actually are involved with between the basic and applied science. Then they could actually find a single point of application that it makes enough sense to basically build a proof of concept So does that make sense?
Very good comment, Mika.
So Ara, what do you think would be the kind of a lowest hanging fruit in terms of valuable company having a kind of a an imminent problem that they would like to solve now?
Yeah, so Mika, I have been obviously much more motivated by the fundamental science, and for a very long time I was very proud of saying that I have made no application, but obviously everything we did years later was incorporated in one way or another in applications. So for example, the single molecule device that I showed, you have to recognize that the touch screen on your cell phone is a fantastically sensitive electrical force measurement tool. So we have higher resolution than that now, right? It's possible to put, so let's say, silver needles underneath a touch screen and to have ultra high resolution. You say, well, the resolution is high enough, so maybe practically it's not valuable. But there will be mechanical, devices where that kind of spatial resolution and sensitivity can be incorporated to make, a much better switch, for example. And that's going to happen, right? So I was not initially at least motivated by a single application, but when we started the concept of, doing an institute, the idea was to push the fundamental science and several applications at the same time, right? Going to directions. So one of them was single molecule devices, right? A device that you can actually practically use in a miniaturized device. The application would be much more above it, right? Every app that you have, underneath it, there are many layers, and ultimately it goes down to the electrical signals between, a few atoms. So devices was, a voltmeter. We have a voltmeter, right? We think we can make any sort of cantilever, etc. That's a factor of 1,000 smaller than existing MEMS devices, which are already being implemented, right? So that's an example of practical direction where we could go. And this needle issue, the Blu-ray CD reader has in it a reader head and a small blue diode laser, which is very inexpensive. Our devices are also miniaturizable, right? So the objective lens in my case is, a micron. It's 100 nanometers, and all it needs is a very weak light source, a diode. So the whole thing can be packaged. It's possible to make the entire microscope I described in, 5 micron, sort of by 5 micron by 5 micron volume. So you can have, this ridiculously high resolution imaging capability, which would be similar to a CD reader, right? It's very similar in the working parts, except that we are in the atomistic near field as opposed to being in the electrostatic near field. So, the disk has to be essentially touching the needle, if you like, right? But huh?
I mean, this reminds me of a lecture I had an invited speaker last week in my center that he's a computational person, but one of his projects had to do with, this concept that's been around for a while now, using DNA as data storage place and retrieval. So you could couple some of these devices with something like that, I suppose, right?
Well, there is DNA origami, etc. People make, use, etc. Ultimately, you can store the information, and then you have to read the information, right?
Yes.
Going in and out. So you need, you can have very high dense coding, but you can't access it, right? if you can now have resolution to access atom by atom, it's more than what you need, maybe. But you have to have that capability, right?
Yeah,
I mean, there is no question that the future is in these directions. This is going to happen. We are a bit early on the spectrum, and I would love to have a multi-pronged approach to applications. But if I'm going to myself be driving one aspect of it, it will be the fundamental part.
yeah.
Okay, so I'm actually, thanking, each participant. So we're kind of, closing at the moment. So I stopped recording. So I still have some questions after that. But I mean, thank you everybody. I mean, it's been wonderful. So from the brain science down to pickle level, what still remains to be answered, and that's a kind of a good thing, is which of the brothers is a better scientist. But so we will have to have a kind of a sequel on that.
No, that's very simple. In the sense of being relevant, it's Vania.
No. I mean, it's a wonderful kind of, brothers. I mean, so happy to have you both here. So I'm stopping the recording now. And